Hi, welcome to this introduction to MetaTrace. This is the software that we are using for data management. It keeps track of your metadata and it connects your metadata to your actual data from your experiments. When you first turn it on, you should be greeted with a screen that looks something like this. So in here, just simply fill in your details. Your email should be your DTU email. Here, I'll just type in this here. And basically, once you create your user, it's going to use your email to generate a unique ID for you for your samples. So if we just go here and create user, you'll be given this access pin. So this is your code that you can give to your collaborators, and then they'll be able to access your data if you let them, basically. Your collaborators will be able to view your data, but with this code, they would then also be able to access and edit your data as well. So we've created a new user here, and we can see I have this sample ID. This is my unique identifier, and this is my first sample or maybe experiment or whatever you choose to use here. And we can see this is record 1 of 1. So this is my own personal database with just one record at the moment. The first steps that you will want to do is set up your profile. And to do that, just go up to options, and in here we can see we can add certain things to our profile. And the first may be a project. So if we click on add project, we can go ahead and just add any project in the list. If your project is not in the list, then you can just ask the administrator about adding your project to the list. So in here, let's just go ahead and add one of these, for an example, and just save changes. Now we have a project associated with our database, and we can also add in sample types as well. So there's a couple of sample types that are by default part of your database, but you can choose to remove these. But then also there's all these other sample types here that you can also choose to add that you might need for your type of experiments that you'll be running. For now though, maybe we can also add this in liquid extract as well, and perhaps well plates. So then we just save changes. Now we have a project and we have our sample types that we need for our experiments as well. So at the moment we have one record in our sample in our database, and if we unlock our database, we're now able to edit these different fields. So for now we'll just click on our project, and we'll see we also have sub-projects for here as well. So we can just select one of those. We can include a sample date, so we can use just say today's date. And now we can also select our sample type. So depending on what sample type we choose, we'll have different inputs that we can actually add to that sample type. So if we have a culture, for example, we will have species, strain name, media type, media composition, description, these sorts of things. And if we just scroll through here, we can see these just update depending on what we are actually looking at. So you'll see that most of these different samples will only have these buttons here available, but if we scroll down to say pure compound, there's additional buttons here that are available for this particular sample type. So what this row of buttons here are data that's associated with this record. So all samples or all records within your database have the ability to add in a folder of data. So this is just a Windows folder, a directory. You can add a directory to each record. This is a PDF, so you can add a PDF to each of your records, and also an image. You can upload an image to your records, which would then replace this here default image with whatever image you choose to upload. Some sample types though also have these additional data types that can be associated with those samples as well. So here we have a pure compound. We can add in a structure to that pure compound. We can add in a UV spectra of that compound. We can add in a fragmentation data to that compound as well. So as you're going through these different sample types, you'll see that some of them will have some of these sort of special data types that can be associated with them. Well plate's another one, so we can actually specify which wells on the well plate have our samples and what those samples are as well. But for now, let's just say we have a culture of some sort, and we can choose our media type, agar. At the moment, there's no media composition, but we'll get back to that. But yeah, perhaps this is the data we need: species 1 and strain 1, and then we can just close our database, and now we have made a culture within our database with this unique ID associated with this culture. Now, for example, if we were to extract this culture, do some sort of chemical extraction, we would want to make a new record, but we'd want to make sure it's associated with this record. So in order to make a new record that's automatically associated with this one, we simply unlock our database again, and this here will create a new record. But since we're making an extract of this particular culture, we would want to use this button instead. It creates a new record from the current. So if we click that, we'll see we open up with this dialog box here. This is the record in which we want to create from, and we only want one replicate in this case, and we want the same sub-project as what we have before, and we're making an analytical, or we can say culture extract in this case. So with that there, we can just create a new record, and it automatically associates a new origin ID with our new sample ID. So this is the sample ID of our culture extract, and this is its origin, this 01 here. So in here again, it will be strain 01, oh sorry, species, that would be species 01. And strain 01. Culture media, again, we'll come back to that. And you can type in the way you did your extraction, how many days that culture was cultivated before the extraction, and other comments as well, of course. So that there, and we can then go ahead and save. That there is a simple culture to extract sort of relationship between your own personal records. One thing here, so if we actually use this button here, it can take us back to any of the origin records that a particular record has. So if we click here, it will list all the origin records, because some records can have multiple origins, or we can show you that later as well. But if we then go here, it will take us back to this origin here. So now that we've made this connection, this series of experiments, the culture we've made, and now we've done an extract, now we can actually get a bit more of an overview of these two samples. If we just go to the table view, we see them, they're both listed here with all the different information that we've put into these different samples. So another thing in MetaTrace is this bar along here, and this is sort of your navigation sample creation bar. This one here, obviously, well, is save. These buttons here allow you to navigate, switch through records like this. A shortcut of doing this is actually if you hold down Control and press left and right arrows, it will do the same thing. That can be useful when you're editing multiple records at the same time. So you could add a comment here, and then if I can even spell comment, and then I can just go Control to the left, it will take me to the previous record, and I'm already in the comments, and I can add in my next comment as well. So that could be quite useful for quickly adding in details. Yeah, so there's navigating through your records. This here is searching your records. So at the moment we don't actually have much data to search through, but in larger databases you might want to simplify your view to say only culture extracts, or maybe your last 10 samples, or something like this. And this is simply done by just, if we go here, we can choose which field we want to actually search on. So sample type, if we go culture extract, now we see we're only record 1 of 1 out of a total of 2. So these would, in a larger database, it would just collapse it down and simplify the viewing of the database, basically. This here simply retrieves those records again, so we get back to our whole database. As we saw before, this button here will take us to origins of the current record, whereas this one here will display records that have come from this current record. So for us, we actually just made a record from this one here. So if we click here, it will search through your database and find all records that you have that originated from this one here. So in this case, obviously it's just the one record, and then we can go to that record. So we can go through forward and backwards through the records based on their associations. This here we've already seen, so there's unlocking and locking your database. A point about that is that as soon as you unlock your database, it's going to assume that you've made some sort of changes to your database. So this here will become ungrayed out. So if we just go ahead and save our database, it's grayed out. As soon as we unlock, it's going to assume that some changes have been made. And that is also true if it remains unlocked as well. So even if we save now, the assumption is that it's still unlocked, it's still editable, so therefore there's still something that you've probably changed that you may want to save as well. So the only way to fully save things is to lock and then save. So that's to make sure your edits that you've made will actually be saved. If it's unlocked and you make edits and you save, it's going to save those edits, but yeah, it's still going to think that there's some editing going on because it's unlocked. Yeah, so that's unlocked. These here are sample creations. So these are new record creations within your own database, these 3 here. This is just creating a new blank record, so we can have a look at that. It just simply gives us a new clean blank record, and we can do whatever we like here. So therefore there is no sort of origin ID in this case. So this is something that's just come from nowhere, essentially. Therefore we also have this here, which is create a new record from the current. So what we saw before, when we have a culture, we extract it, it becomes an extract. You could also use this to split a record into multiple. So if you have a culture and maybe you make 3 different extractions on it, then you could say make 3 replicates from this current one as well. So I can just do a quick demo of that. If we go back to the start here, from our culture, we can go here and we can go ahead and say culture extract, and then we can just say replicate, 3 replicates. And these would have perhaps three different extraction procedures based on whatever our different methods of extraction would be. So if we go ahead and create records there, we can now see we've got up to now record number 6, still with that same origin, the same species and strain as well. If we go to our table view, we can see these 3 here. So this is the one that had no origin that we just made before, and now these are the 3 additional extracts from the sample here. Yeah, so this is creating these links between your different sample types. Another example would be, say, a fractionation of an extract. So you have a crude extract, and maybe you're separating that extract into multiple other extracts through fractionation. We can do this here as well. So say if we take sample number 4 here, we click on this button here, and maybe we make, say, 8 fractions out of here. Do we have even fraction? Yep, we have fractions here. Fraction information, so we can say fraction number, and then if we do this, rep hashtag, wherever the hashtag is gone, nope, yeah, and if we do this, when we create these fractions, it will replace this this symbol here, these square brackets rep and hashtag, with a 1, 2, 3 up to 8, depending on how many replicates we make. We can say fractionation of sample. And then if we just go ahead and create new records, we can see we now have fraction number 8, 7, 6, 5, 4, 3, 2, and 1. So, and back to the table view, we can see these are our fractions that we made from number 4, which was this extract here. So we have a culture, which we made extract. From that extract, we then had these fractions created. Also, what we can do is merge samples as well, merge records into one. So in, say, this example, we may have fractionated an extract. Maybe a handful of those fractions actually contain the same compounds, so we want to merge those, pull those extracts together. And we can simply do that here as well by just clicking this button here. So this is the merging of records. It's defaulted with 14 because that's the sample we're on, but maybe it's not 14 we wanted, maybe it was 11 and 12, so simply just remove that one, go add 11 and 12, and this could be another analytical extract or maybe a fraction as well. Probably an analytical extract. So the sample type you use is up to you, and some might sound similar, but they'll have different entries, so you can just use the one that makes the most sense to you. If there's a sample type that you think is missing, also just contact me and we can add in new sample types as well. And just one replicate in this sense because we're just taking these two fractions and pulling them into one sample. So we do create sample, and now we see we have this new analytical extract, and we have, in this case, 2 origins. So now if we go back to, let's just put in the same subobject. If we go back to the table view, we see now we have this analytical extract that has come from 11 and 12, which was these 2 samples here, which came from this extract number 4, which came from this culture number 1. So we're sort of building up this idea of how different experiments, different samples that we're dealing with in the lab, they're all sort of somehow interconnected, and we can build up our procedure that we did to get to an endpoint based on all of those steps in between. And in a lot of cases, it may seem a little bit tedious, but this is a fairly simplified way of just making sure we can keep track of these processes. Particularly important when we're collaborating with other people as well. So if you give someone a culture and then they can view in your records exactly all the information about that culture, where it came from, or same with an extract or even a purified sample, they can then go back and say, oh, this is how that sample was purified, or this is the extraction procedure they used on that particular sample. So that was these 3 buttons here explained. The other one, this is for adding and removing origin records. So here you can see we have 11 and 12 are the origins here, but perhaps we actually made a mistake. It wasn't actually 11, it was 12 and 13 that we wanted. So we can simply just click on here, we can remove 11 and then add 13, and then save changes. So now it is actually 12 and 13. So we've corrected this sort of linking of these different records. If you open this here and you see actually it's not a full list on this side, it could just be because you haven't saved your database. So we've made a lot of records, but they won't be populated within this list until we've actually saved our database. And if we, maybe I can do an example here. So if we just create a new blank record, we now have 16. And if we go back and we want to add 16 to here, for example, if we look in here, it's not in the list. But as soon as we save and then come back here, it should be there. Exactly. So the idea around these creating records and things, you'll notice that there isn't actually a remove or delete record. And the reason for this is that we have to make sure we can't delete records, because if we make a connection, say, between 12 and 14. If at some point we, for some reason, delete 12, then 14 no longer really makes sense because it came from 12. So we sort of lose that connection. Another thing is that, say, if we deleted a record, then those unique IDs would, I mean, you could have an idea with this, all the records higher than the one we deleted, their ID would then shift lower. So it would complete that sequence of record number 1 to record number at the end, basically. So we have to maintain the unique IDs for these samples. So we maintain these sort of connections between all our samples. We've only really looked at a very simple example of connecting these different records, but these chains between records can, in theory, be very, very huge. So, for example, if you could imagine someone goes out into nature and picks up an environmental sample that's got one ID. From that environmental sample, they isolate a strain through multiple culturings. So there's a couple more IDs there. They deposit that strain into a library. Then someone downloads that strain to make a culture. From that culture, they extract. So this chain of these origins can become very, very long. So we don't want to be in a situation where we could delete a origin somewhere in the middle there by deleting a record and then lose this continuation of how we got from A to B, basically, or A to Z. So that's this button here, and then we have these two here, and at the moment, if we click on these, we'll come up with this warning here. We must first load a facility in order to upload records to a library. And it's the same for here as well. So these buttons are for special cases when we have these a particular type of sample or record that we want to upload to a library, a facility, so other people can have access to it. And we'll come into that very shortly. These here are downloads. So these are uploading to a library, these are downloading from a library. And since we're not in a library right now, we can't actually use these buttons, but we will explain these 4 buttons very soon. And here is just a very simple chat function, so you can send notifications to other users asking to be collaborators or whatever. So yeah, that's basically the standard buttons, navigations and things within MetaTrace. So another thing would be, what we could do is add a collaborator to our database, which is simply going up through here, adding a collaborator. When you add a collaborator, they will then be able to view your records for particular projects that you give them access to. They won't be able to edit those records unless you give them your PIN that we saw earlier. So adding someone as a collaborator, they can view yours. You cannot view theirs until they add you as a collaborator. Once you've added each other as collaborator, if someone's added you as collaborator, then you can view their records via this button here. And at the moment we have no collaborators, so we don't, okay, actually, apparently we do have a lot of collaborators. So we can actually view some of these records that this user is a collaborator to. And we can save the changes that we've made here. And now we can see we're in this sort of more green version of MetaTrace. This green is indicating that we're now viewing a collaborator's database rather than our own. And we can see if we try to unlock here, we'll have to enter a password to unlock. That'll be that PIN, whatever PIN it is for this particular database. It doesn't simply just unlock automatically like we saw before. Now though, we can see we still have these upload buttons, which we still won't be able to use because we don't have a facility open. But we do have these download buttons. So these downloads we can download from facility libraries, or we can download these samples to our own personal library. So for example, if someone makes a culture and we want to extract it, we can go into their database, find that culture, and then download it into our database. So for example, we can just, in this example, just pretend that this is in a culture, for example. We can click this button here and we can download it into our own personal database as an extract and then create sample. Now we've got a new record in our database and its origin ID is our Collaborators database here. So that's sort of how this downloading works with these buttons here. This is downloading a single sample to your database, or you can actually do multiple downloads at the same time. The only difference between these two is then this here is actually merging multiple. So for example, if you're in a strain library and there's 7 different strains and you want to make a synthetic community experiment, then you could download those 7 strains into one culture in your own personal database. So that's those download buttons. We'll still come back to these uploads, but first we can take a look at this button here. So this is where we need to access our own database. This is when we want to access our Collaborators databases. This here is a separate set of databases that are data systems. So for the example we'll look at is, say, LCMS data. So you may make an extract in your own database and then you run it on a particular mass spectrometer. That mass spectrometer would be in the list here. And that way we can link the actual real data to our metadata that we have in MetaTrace. So if we click here, we'll get a list of different data systems that we have. And for here, we'll look into Galadriel, which is one of our LCMS systems that we have. And we can click open here. And now we have this sort of teal color, I guess it's teal, I'm not sure. But yeah, this is indicating that we're now in a data system database rather than a collaborator or our own personal databases. In here, we can see these are, each record in here is a batch is a work list, more or less. So this is a set of samples that were run on that particular day. So if we look at, say, just one of these, we can see this was run on this date here, and these were the samples that were run. So this work list within this batch had these samples run. So it links, and these were for example extracts in a database, in the database unique to here. So it links our actual samples, our actual sample data, to our metadata that we're creating in MetaTrace. And if we want to find that data, then we can simply just click this button here, and this will open our data file. And this will be seen, once it pops up, this will be seen as in right here. So these are the data files that we've found here. So this links our physical data, our raw data, with our MetaTrace. So now we're sort of expanding this sort of chain of information regarding our experiments from culture to extract being run on our actual data system. So that's the basics around that sort of workflow. If we go back here to our own database, we still haven't really covered these buttons here. The thing about these is that these are associated with facilities, and a facility is maybe a research group, or it could be, in our case, the metabolomics core facility. It's any sort of entity in which multiple databases are needed for the common good, in a sense. So it could be a strain library. It could be a list of publications. It could be something like a metabolite sample catalog. It could be a list of analytical columns. It could be primers. It could be plasmids, along these sort of lines. So, but at the moment, as we're looking at our MetaTrace right now, we don't have any facilities open, so we can't actually use these buttons just yet. But if we go into facility, we can select a facility here. So by selecting a facility, we'll get a list of the different facilities that are available. And if we go down to, say, the metabolomics core, we'll have to enter a PIN. Every facility will have a PIN that you have to enter to be able to access it. It would then also have a separate PIN that you will need if you want to edit that facility. But for here, we can just type in the PIN for the metabolomics core. And now all of a sudden you see we have many more different options down the side here. And these are the different libraries that are within the metabolomics core facility. We also see that we now have metabolomics core up here. This is indicating that we're logged into the metabolomics core facility. Now it's quite useful if you know there's a particular facility that you're going to be using a lot, then you might want to go into facility. Actually, you have to unlock your database. Then go to facility and you can set current as default. So if we do that, it just tells us that we have to, yeah, we've updated our default facility. So now that's quite useful. So whenever we open up MetaTrace, it will automatically also open up the metabolomics core facility every time. So we don't have to go back in here, open up the facility, type in the PIN to get those libraries to appear here. So we're opening up the facility. We're actually still in our own database though, under My Samples. But now we have the option of looking into more of these different libraries that are associated with the metabolomics core. For example, in here we have a list of different labels, for example, for chemical waste, acetonitrile, different solvents and things that we use. This is just our test database, but in the actual database that we have live, we also have PDFs in here and images of those labels, so we can open up the PDF and print them out. Quite a useful feature, I think that would be in a lot of facilities, is a strain library. So if we click View Strain Database, we'll see it opens up these additional databases here, and these are different types of strain libraries. These are some of the options. So we have an algal database, a bacterial database, a fungal database, a virus database, a plasmid host database, and also a cell line database. But I think for now, we'll just maybe try out the fungal database here. And now we see we have 1 of 13 different strains that we have in the strain database. Again, this is just a test, a dummy sort of database, so none of this really makes sense, but it would work in a similar fashion in the actual case. So for this example, maybe we can find something that maybe makes a little bit more sense. Okay, at least this now species is associated with species and strain. So then in here, we can use these download buttons. So maybe this is a fungal strain that we want to make a culture of. So we can just simply click this button here, and maybe we want to make 3 replicates of this culture, and we're doing it under the subproject of apples. And we can select the media type, and now in media composition, we actually have a list of different media. We didn't see this before, because this is pulling media information from our current facility that we have open. The benefit here is that it will be unique to the facility that you're working with. So it's not going to be a list of 100 different media types that you have to search through, because each facility will probably have their own specialized medias that they work with. So in this case, we will just say use SIR, and making 3 replicas of this particular fungal strain, and we just create records. And it's filled in all this data. We now have the strain that's associated with this culture. And if we look in the table view, we can see it's made these 3 replicates here of those cultures. So this is using this facility to make our cultures. It's a much quicker way of filling in this data and making those connections. We don't have to type anything really. It's just a matter of clicking a couple of buttons. Now we have 3 new unique IDs for these 3 cultures that we have just created. So these 3 here. Yeah, and if we just go back to the one that, so we're back into our own personal database. We downloaded that into our personal database. If we look back around here again, so it's now since we created these new records, we now need to save those records that we've just created. If we go back to here, so we can actually look at the different media types that are associated with the metabolomics core facility. If we click here, then we have a list of these different media types, and these are the media types that we see in that list when we create those new cultures from the library. Also in here we have different extraction procedures. Again, this is just a dummy database, but in the live database we'll actually have PDFs that you can open up within MetaTrace that actually explain, it's the SOP for that particular extraction procedure. There's other things like primers you can have. Also, there's these are columns, so these are analytical columns, preparative columns, GC columns. There's also publications. So the difference here, you might be seeing that these are yellow, whereas these are more of an orange color. So the difference is these orange colors are records that can be connected to other records. So like a strain can be connected to a culture. This is metabolites, so a particular metabolite sample can be used in a bioassay, for example. A plasmid can be introduced to a plasmid host. An environmental sample, a strain can be isolated from, etc. Whereas these are more references. So these are things that we can refer to, but their individual records don't actually link to new records. Extractions, database types, a particular primer sequence, these analytical columns and safety labels and things like this. So these are sort of useful resources, whereas these can contribute directly to those experiments. So with these, perhaps you can also start to see how these connections can all come together. So when we're going from a strain to a culture to an extract, and then that can come all the way back around to a publication. So that way we can get a whole overview. Say if we're starting at the publication, we can get a list of origins. So now we've looked at downloading records using these buttons from Collaborators and from these facilities. But what we should also look at is uploading one of our own personal database records to a facility. Perhaps that could be something like we have an environmental sample that we want to include into the facility itself. So if we're here, we could create a new sample. Do we have an environmental sample here? We don't have an environmental sample, so we just need to simply go to add sample types, environmental sample, add that in there, and now we can go environmental sample, and we can choose our project that we're using, probably soil, makes sense. If we're using a soil sample from the environment, short description, soil sample. And we can add in this additional information here as well. I don't know, how do you do GPS coordinates? North, south, something like that, I don't know. Collector, me, date. That kind of makes sense. Denmark. So now we just want to upload this to our facility library, the materials library within the metabolomics core. So first we just have to make sure we save, because this is a new record, and then we click this upload button here. And now we need to select the library to which we want to upload, which would be this material sample. And we'll see this record 21 is actually the only record within our database that is allowed to be uploaded to a material sample library within the metabolomics core. So only particular sample types can be uploaded into libraries. So for example, you wouldn't be able to upload an extract into a strain library, or say a culture into a chemical compound library. But you could upload, say, a culture to a strain library. So you've isolated a strain via culturing, you could upload that as a new strain. In this case, we have an environmental sample that we can upload into our material sample library. So we need to just simply add that here. And we see this is now updated with our sampling location. So some of these fields will automatically update. If these record values are identical to these record values, then they should automatically update. So perhaps what we can do is actually update the way this is called. Instead of just GPS, we could do GPS location of isolation. And then it would also copy across our GPS information. Not that it's particularly interesting. Yep, so if we go create new records, do we want to save the changes we made into our current database? We can just go yes. And now it's taking us to the material sample library within the metabolomics core. Since we're now trying to edit the library within the metabolomics core, we have to put in the password for editing that facility, which is different than the one we saw before. We were just trying to view the facility. So let's just see if I remember. Yes, I did. So now we have a new sample within the environmental sample in the material sample database, which has the origin of our previous record. So you can imagine this could be, here we looked at an environmental sample. This could also be, say, cultures that produced a new strain. It could be a new purified compound that you want to add to the metabolite library. It could be the resulting experiment that went to a publication, so you could upload it into a publication. It could be, any of these sort of things. So that was one example there. If we go back now, it will ask us to save. Yeah, we can just save. Another one is this here. So this is merging multiple records into one. This is probably wouldn't be used as commonly because there's not many situations where you're merging multiple things into a library within the database, within a facility. So we wouldn't merge multiple cultures of different strains into one strain, for example. So if we look here, there's not many. So there's metabolite samples. So in this case, you might have multiple fractions that have the exact same purified sample, same metabolite, and you might just merge those together into one entry into the library, into the metabolite sample library. And of course publications. So you can always add multiple samples, multiple records into a publication. So to that, for example, say we did this experiment, we used this material sample, and I think this was a fraction. We use this fraction in a particular publication, and here we can choose to create a new publication, or we can append to an existing publication. So these are all the different publications that we have in the metabolomics core in this test database. So we could choose to, in addition, add these to an existing publication, or we could, so these are just obviously test database stuff. So we could add it to this, our second publication within the database. So it should add 12 and 21 to this already origin list here. So create new. Again, we just have to put in the password. And now it's added these additional origins into this publication. So now we've covered all of these buttons along here. And we've also looked into the facilities, we looked into the data systems, and also our own personal databases and our collaborators databases. And that's more or less this sort of functioning of within MetaTrace. But a nice thing about having these unique IDs is that we can also use these unique IDs in the lab to actually label our samples. So in order to do that, we just need to go to File, Create Labels, and make HPLC or Eppendorf labels. So if we click on HPLC, we will see we get this PDF, and it will be a PDF of every record within our database. And if we scroll through, we can see these are the different fractions and cultures and environmental samples that we've just made now. And each one will come with a data matrix code as well. So this actually just simply encodes this ID as well. So if you've got a 2D barcode scanner, you can scan your samples and it will automatically write out whatever your ID is for that particular sample. So this is very useful if you're going to run a whole lot of samples on some sort of instrument and you're making a work list. You can just go through and scan them all. Yeah, so it's in a particular format that we are using for our sampling, for our labeling, and then you just simply print these labels. So, but most of the time you obviously won't want to print the entire database, but the PDF will be your entire database. So you just need to choose which pages you want to print basically, and the page number is equivalent to your ID number as well. So page 12 is sample 12. So you can just say print 12 to 20 or whatever you like. And same goes with the Eppendorf labels as well. So those the labels. You can also in here, you can export your database as a CSV. Depending on the current record that you have open, if it's got any of these associated data on it, you can also export that data that's associated with your particular record that you happen to be on. Here we're on record number 1. It has no associated data, so we can't export any of that data. Yeah, but you can also export it as CSV, but also as a network graph. So this will export it, your entire database or multiple databases, depending on what you choose, as a graph file that you can open in software like Cytoscape. And what that will do is each of your records, or each of the records that you choose, depending on the databases you choose, will be a node, and there'll be connections from those nodes to other nodes. So there'll be connections from one record to another record, depending on their origin IDs, as we've been working through. So it can build up a network of your experiment, basically, going from, say, a strain to a culture to an extract to a whatever you're doing. Yeah, and the record, there's just some basic stuff. So editing your database is the same as unlocking your database over here that we went through. And then here are some additional little things that probably aren't used so much, but append record is just the same as using this plus button. Append multiple records, it just allows you to put in multiple new records all at once. Duplicate records, so that can be actually kind of useful if you've made a record and actually you just want to basically copy and paste that into a new record. Go to whatever record that is, click duplicate, or just copy and paste it basically. If you forget your pins, you can see them all in here. You can look at my database pins. If you have access to one of these, if you're the administrator to one of these facilities, you can also view the facility access pins. You can also change your pins as well if you like. You can change database admin. So that means, say for example, you might be leaving and you need to hand your database over to someone else. Here you can change who is your database owner. Yeah, and you can restore your database as well. So if you, like we said before, you can't delete a record, but if you find you make a mistake, you can unlock your database and then restore it to one of your previous save points. And I think it saves up to maybe 10 or 20 or so. It saves along the way. Yeah, so that's more or less it. I've gone through all of these. Add and remove super user, that's more for if you're a facility manager and you want and by adding a super user to a facility, then they don't have to put in the code every time they want to view or edit the facility. Edit labels is actually quite good to know. So in here, we can see that we have barcodes turned on by default, but these are the entries that we're going to see on our label. By default, we'll always see the unique ID along the top and along the side, but these are the other entries that we can choose to have or not have. And if we're looking at these entry numbers, they are simply as listed. So 1, 2, 3, 4, 5, all the way down, where 15 is the comments. And record date, it's going to look through these different record entry names and look for something that has date in it. So here, creation date. So this will be added also to the label. You may want to actually use today's date instead of the current date. So you can remove that and then add in today's date. So we'll just put in whatever the date is that you're printing those labels. So for example, you could be putting your metadata in one day, and then the next day you actually go into the lab to do it. Then maybe you'd prefer to have today's date than the record date. Yeah, the one thing you have to be sure of is that there's always 6 entries in here. If there's 5, if there's 3 or whatever, it's going to tell you, hey, you need 6. If you put 7, it's going to tell you you need 6 before you can save it. So just make sure there's always 6. And then you can just simply save that. Yeah, and then facilities, we select facility. You can also remove that default facility. So before we added the metabolomics core as the default, we can also remove it. And these here are if you're a facility manager. So you can change the admin, you can look at the pins and all that sort of thing as well. And you can add different libraries, resources to that facility. So you can sort of simplify your facility view. If you know your particular facility is never going to be working with cell lines or virus libraries, then you can remove those as options within the strain libraries, just to simplify the overview within your facility. So all these are optional. Systems, this here is also something for the administrator of the MetaTrace system. So we can add new data systems. So if you have a new, say, HPLC, a new mass spectrometer that's producing data, we can add that new system into these data systems here. So then that data can then be linked back to the metadata. But yeah, that's about all. This has been an introduction to MetaTrace. There are some other little things that we could go into, but I think maybe we'll just leave it for this for now. Thanks.